Thank you for organizing this conference again. I mean, this is really a very timely summary of what we have done, I would say, since we started Deuteriums. It's 2019, our last meeting, when we started kind of naming this field because it was necessary to summarize all the medical applications that are coming out now with this new approach. And we started back then when Gabor was a student with his note, which is very famous for many reasons. But what is really timely nowadays is that he's talking about protons or hydrogens. He talks about electrons, which was actually from Dr. Albert Szent-György's television conversation, Here is Hungary. And he talks about the size of the protons or the size of the atom and the protons. And this now has major applications in the tunnels or in the tunneling process. Now, this was exactly 50 years ago. We are here and we are still here. And we are here because of this. It's kind of easy to name or figure out when we started. Exactly 50 years ago when Gabor as a student summarized at that time. We're going to come back to this note simply because it's very significant. And this is a handwritten note in Hungarian, so I didn't want to show it. But the English translation, which Gabor gave me a few years ago when we started presenting. Now, to understand why Gabor's note is so important and what Deutenomics is, especially medical Deutenomics, we have to go to a paper which is a very recent paper, and it talks about metabolic profiles and transitions required for long-distance avian migration. And this is where mitochondria are truly challenged. This is when metabolism is taken to the ultimate boundaries and constraints. And there are very, very important applications of what we learn from this model. Now, we actually talk about this bird. So let's do a flight lesson here, just to cover Deuteronomics. It's called a Hudsonian godwit, Limosa hemastica. Just if biologist, Gabor is a biologist, so I have to use the Latin nomenclature as well. But what they do is actually very intriguing because they fly from Alaska all the way down to New Zealand or Tasmania, and they tagged a few of these birds, and what they learned is that they either fly to New Zealand, or if they have high headwinds, then they turn and land on other islands, which means that this is really a very challenging biological principle. And to understand mitochondria and what happens in mitochondria, all the flux-related, proton size-related issues, we have to understand this migration pattern, which they do twice a year. And they traced one of these godwit. It's like a half a kilogram bird called B6 as a code name, and flew nearly 13,580 kilometers in 11 days in October 22. And if you calculate the average speed, that's about 51.4 kilometers per hour. And it's a nonstop flight because this is actually a land bird. So they can't really stop halfway or just rest or drink some or sleep some, or this is really challenging. And I think this is the way to understand, if we go through this whole process, how significant Gabor's note is and why we are here today. Now, and these are just calculations that I did. They burn about 130 grams of fat based on how much weight they lose. And it's a continuous flight, we know. And they depart this type of bird, or this version of it, which is the striped tail. They actually start out with 280 grams, and they land with 250 grams. So they burn, that's their fuel. They burn about 130 grams of fat. And this is their cycle. You can see here the fattening part of their life cycle before they migrate, and then they do it again. And they do this twice a year. So I would say even a jumbo jet from Alaska has to land in Tokyo for refueling. I flew a few times from Los Angeles to Australia, and it was even for a passenger jet, is not an easy task. They have to schedule for refueling. Not this bird. This bird is able to fly. This is their weight. It's now published in scientific reports, so with the link, Gabor is going to share, I guess, the PDF. But this is practically the weight difference. So it's 130 grams of fat, and there is a fattening they prepare by accumulating fat. And this is actually 1,170 kilocalories of energy if you use the 1 gram and 9 kilocalories fat. Gabor mentioned this yesterday, this data as well. We use these calculations. This is actually about $30 worth of pork crackers, and it's about 1.4 kilograms of this for us to cover by weight of how much energy this is. And they mobilize these 3 fatty acids in particular. What is the phytanic, docosahexaenoic, and myristic acid, which is a 14 hydrocarbon or fatty acid. These are ketones, ketone fuels. One is C20, 40 protons or hydrogens, 28 protons, hydrogens, and 32 protons and hydrogens. This is less saturated. These 2 guys are saturated long-chain animal source fatty acids because they are land-based birds and they eat on the shore. They eat snails and worms and those kind of things. They are not plant eaters. And these fatty acids, if you calculate the grams, how much they burn, they provide 293 kilocalories of mechanical energy at 25% efficiency rate. This is what we use in ornithology. So anyone can tell that Houston, we have a problem because it seems that the fuel amount that they burn does not cover what they need to cover the trip. So we're going to have to find some other important critical roles of what mitochondria do and how they accomplish these migration. Newton's third law of motion is practically these birds have to generate forward thrust that is equal to the air resistance or the drag, and 11 days to cover 13,500 kilometers over the Pacific Ocean using 0.26 Newton as the drag that they have to work against. And this is actually a calculation from a presentation from the University of Hawaii. This is an airplane, how they fly. There is a forward and there is a thrust, there is a drag, and there is a lift, and there is gravitation, there's weight. So practically these animals have to balance these out. And apparently when we calculate the work, which is the force multiplied by the displacement or the distance, actually we come up with a 838 kilocalorie values, which is multiple times of what the animals burn in the form of fat. So there's a question here, how is possible? And this is really interesting in the sense that it's actually short of, they only can cover with the amount of fat, if we just do the calculations, just the drag, nothing else, no lift, no heating these little animals, just the mechanical force or work they do. They can only cover 4,700 kilometers, and they are short of 537 kilocalories. That means that they cannot cover 8,700 kilometers of their trip. And for that reason, they might just fall into the ocean mid-migration. So here's the challenge. Houston, we have a problem. And if we rethink mitochondrial metabolism, and if we go back to Gabor's note, we figured out 50 years ago, actually, he solved this problem. Simply because he mentions the size of the proton, and he talked about, not directly, about collective electron and proton tunneling. The size and the mass of the proton is critical to understand this process, and the input from other energy sources. Now, don't give me those looks, okay? At that time, I was only 14 years old. That's why I didn't write this message, because I couldn't stay up late. My parents didn't allow me, because this was actually, I think it was late at night, around 10 o'clock, this conversation with Dr. Szentgyörgyi, if I remember correctly. And there was actually, they repeated it like years later. But actually this coupling electron protons and naming the atomic and the nuclear size was practically a revolutionary idea, simply because that's what it is. That's what really matters. That's what Deutenomics is, after all. And Gabor was the first who couldn't connect, simply because we didn't know nanomotors, we didn't know proton pumps, we didn't know a lot of things, but actually these are from the atomic space. This is, there are several questions. Did evolution miss something here? I mean, it's hard to imagine a species that would survive by kind of walking in Alaska and like, hey guys, there is a 14,000 kilometers island somewhere. We couldn't make it 14,100 kilometers, but 14,000 will happen, so let's just go. Apparently, that's not an evolutionary idea. I would kind of wonder if a flying Darwin would actually depart or undertake this journey with these calculations, or should we just take another look at light waters? It's very important, deuterium depleted water, what mitochondria produces. Light waters, mitochondrial collective electron tunneling and proton tunneling as an energetic solution, uncompromised by deuterons. This is the name of the game here, which is really, really a fascinating story. Here is one of these nanomotors, kind of an electromagnetic scheme of it. This inherent collective proton tunnel pattern is very important when we generate ATP, but ATP is not as important as water, and we're going to get into those details, deuterium depleted water. And we need to kind of go through a few very basic principles. These ATPs, nanomotors, or complex fives, are, by their very nature, inherent unidirectional tunnels for individual protons in a collective electron-coupled manner. And water production is the end of this whole result. Water production is what moves this entire system. And due to the above, mitochondrial proton tunnels cannot adapt to deuterium whatsoever. And this is because of their genetic and also proteomics imprint. That's why it's called Deuteronomics, because it comes from those layers and those levels of understanding biology. Here's a nanomode, here's a mitochondria. And here we're going to see these complexes, complex 1, 2, cytochrome C and cytochrome oxidase. Complex 5 is here. And what really matters here is the proton, which are these white little dots. As they reach to the surface of this ATP synthase nanomotor, they only have a tunnel, and this is what we call a proton tunnel. They only have a tunnel that is the size of a proton. And actually, this proton attaching to this nanomotor is entering the mitochondrial matrix. On the other side, there is a longer tube that helps the proton to roll into the matrix where oxygen is waiting on complex 4. And this oxygen will make water with this proton. And water is recycled to fumarate hydratase, which Albert Szent-Györgyi got the other motivation for his Nobel Prize, the catalysis of fumaric acid, which is part of the Krebs-Szent-Györgyi cycle. But anyhow, in the meantime, ATP is getting synthesized. But really, truly, what matters here is the proton, because, and Esther did speed up this process because they rotate 100,000 rotations per minute, some nanomotors. If deuterium comes, which is this red dot here, it will stop passing through this whole nanomotor system, these proton tunnels, and it breaks these nanomotors. Dr. Olgun just published a paper which clarifies this entire process more with more clarifications. Now, the important part here is that the same structure of ATP as nanomotors are present in all kingdoms of life. There is no prototypes of this. It's the same in every species, every living organism. So there is no evolution of this nanomotor system, and human mitochondria make sure that nothing changes the size of these proton tunnels. And that's practically through this mitochondrial DNA. It has 16,500 base pairs, 37 genes, and 13 proteins, and those 13 proteins are all responsible for proton tunnels. Here I listed them in this red square, complex 1, 3, 4, and the nanomotor rotating part. These are all harvesting hydrogen or deuterium from food. And deuterium cannot fit in there. None of them are designed to let deuterium pass this system. So practically, when we look at the nanomotors, deuterium is guarded not only through the nanomotor, but also through the NADH oxidase or these oxidoreductase systems that are supposed to harvest protons only because deuterons will not fit through there. So this is a clear shot here. And this is why actually we now call this medical deuteronomics as what it is, because mitochondrial proton tunneling is the key. Electron-coupled proton tunneling is the key to understand at least what fuels the system. Now, proteins that are in this slide is important simply because proteins that have not been and cannot be altered by evolution or adaptation, as these are the mitochondrial proton transferring proteins, because these are actually monoallelic clones of maternal inheritance, meaning that all we are here and all the humans who live on this planet, they only inherited proton tunnels from their moms. And this is because what happens at the time of, like, when you become pregnant, these mitochondria-encoded proteins gate out deuterons, and the moms make sure the maternal egg makes sure there are no deuterons that can make it through the system, meaning that in the time of and this is a very important highlight of matrix water, that's the characteristics of it, it's the most deuterium depleted water source on this planet. It's probably deuterium free. We don't have direct data with that. MRI studies, now we are clarifying some of these details, but we believe that when this gets diluted by other various sources of water and food, then we have a deutenome, which is the deuterium map of any human being or any living species. And we now believe that actually species is defined by their deutenomes. And water is recycled in metabolism. We call reductive carboxylation. I'm not going to talk about this process, but this source of deuterium-free water or very low deuterium water in the matrix provides the genetic stability, and once this system is compromised, like Gabor was telling yesterday about compromised mitochondrial functions in tumor cells, you actually disrupt this internal compartmentalized deuterium depletion, fractionation, discrimination process. Now, the female gamete, which is the egg, would kill the sperms or the male egg's mitochondria. So there is no allelic variation in proton tunneling. There's no evolution in this process. So it's very critical to understand this, the importance of this. And if you guys study histology, this was, I would say, one of the most important kind of information to know about the spontaneous abortion process in gynecology. Simply when this process is not happening perfectly, then the egg terminates itself. I'm not going to talk about more details. Humans and all other species were given mitochondria as ketogenic, low deuterium, natural fat-powered nanocompartments, and this is how these birds can fly. And we'll cover that in a little bit. And human mitochondria cannot adapt to high deuterium containing refined sugars and or high toxin plant-based or poison nutrients, glyphosate, for example. Dr. Senef is going to talk about it. So there's a lot of connection here that can be connected with other talks before my talk. And deuterium depletion is a shared biological responsibility or deuterium regulation. If somebody has no access to deuterium depleted water, then your biology has to dissolve, solve this problem. Among prokaryotes, gut microbes we collect or food deuterium end up, and eukaryote epithelial cells and their intracellular compartmentalized proton exchange enzyme reaction architectures by sunlight. And this is what makes this little bird to be able to fly, even though there are headwinds at the end of the game. Simply because what they do is you can move out this obstacle simply. We all have our tricks, and their trick is practically and this is actually very important, sunlight makes oxygen and hydrogen for our mitochondria. These are Nature papers. You can click on these when you have time, but this is actually, they have sunlight over the ocean. We know that. There's nothing else. They can't land for food, they can't rest, they cannot do anything other than just go to a higher altitude, which they do. And they actually soak up more sunlight. And what they do is, just like in chlorophyll, melanin is able to break water. 2 water molecules are broken up into 2 hydrogen and 1 oxygen gas compartment. And this gas, when it's formulated back to water, again, mitochondrial water, 285 kilojoule per energy is produced per mole. If it's hydrogen peroxide in peroxisomes, then 187 kilojoule per mole energy is produced. And when it comes to ATP rotating, actually ATP synthase is nothing else, just a roadblock that they have to jump over, let's say. And power it, but practically it's only 20 kilojoule per mole. So the most energy comes from water formation in mitochondria. It's not ATP. I know everybody is talking about ATP, but it's not what it is. It's actually the water formation process. And I kind of circled or squared these, very important. We published this paper with Dr. Seneff, and there are more calculations to this process. These are, I would say, the most medically positioned papers of Gábor's work. And these are very fundamental to understand the system that we have been talking about. And this is a peroxisome, and this is a mitochondria. And they actually crosstalk each other through NADH, proton tunneling. Again, this is where fatty acids get oxidized, and heat and hydrogen peroxide is produced. And this is the mitochondria where catalase can produce metabolic water, and this metabolic water can be split by melanin and the oxygen and the hydrogen gas recycled and make water again. And this is a sophisticated system because now you don't have to kind of stretch it out or plan. If you're a birdie, you just eat up with fat, build up enough fat, and you just supply a little bit of water on top of this. But most of the energy comes through this atomic proton tunneling electron-coupled collective process, which we actually talked about with Roman many times. And this is why we brought our PNAS paper, just to give it a biological adaptive mechanism and edge. And this is actually one of the most, I'm a religious person. I think God sent me this paper as an editor, so I had to look at it more. I did these calculations. I didn't send this to the authors because I didn't want to freak him out, but practically this is part of it. The strength, endurance, and this is just a summary of what I would like to cover here. Sunlight, melanin migration provides for us too. Don't misunderstand or don't try to kind of skip it, but we are in the same situation as these migrating birds. And truly, we have our own tricks, but practically, we don't have to run to Tasmania twice a year. And so this is not the same kind of challenge what these birds face, but in health, in our case, is from the unit that is deuterium-free recycling of the protons from food and oxygen from air through single proton tunnels during metabolic water formation and recycling. So this is practically the essence of life. If you have no oxygen, no question, you have maybe a minute to live, and then if ambulance can bring you back through these masks, that's fine. But the proton pool is a little bit like more reserved from our fat sources and stuff, but actually this water production is life. That's what it is. If you leave this process interrupted for a very short period of time, we know that's practically the end of life. And it's true of all species. And this is the only capability that we let our offsprings or our children to carry on with this way of producing life. I think this is why the eternal water of life is so intense in old scriptures. By the way, it's also Genesis, or the first book of Moses, that Adam has to name the animals just to kind of take care of them. And this is what Deuteronomics says. It takes care of this part of biology, the water production system in the deuterium depletion system. And this is the I would say, one of the most important and critical understanding of how this little guy can do this trip twice a year, even when headwind comes, and there's no food around and there's water, they would die definitely. So they actually know this better. And somehow 50 years ago, Gábor knew this better. it is as interesting as it is. Thank you, Gábor, for being such a good eminent student back then. And surely this complex 4, this oxygen harvesting, water producing system with the nanomotors that gate out deuterons because of these proton tunnels. This collective proton tunneling, electron-coupled collective proton tunneling is the essence of health and life and endurance, as we were covering here in this talk. So in this paper, in the discussion, the authors were making a remark, and I'm going to read this. So it spells out the significance of these proton tunnels. Although our metabolomic approach did not directly assess isotope distributions or deuterium enrichment patterns, recent work has proposed that carbohydrate remodeling towards unsaturated and saturated fatty acid biosynthesis may contribute to mitochondrial protection during high energy demand states through selective deuterium depletion through and within metabolic pathways. And I think, and they cite 2 papers. One is Dr. Ogun's paper, the first one that showed how deuterium or deuterated water destroys these nanomotors. And they just published a new paper where they actually show that the unit C on those rotors, on those nanomotors, their conformation is determined by deuterium. So to form a proton tunnel where protons fit through, it actually depends on the deuterium water proton ratio of these nanomotors, of the surrounding water, structured water and surface water that we get from the environment, from food and produce in mitochondria. And after all, understanding this process is, I would say, the future of, I covered where we started 50 years ago, and the future is practically how much understanding this process we can introduce this from elementary school education, religious, theological, natural science approaches, high school, college, and Kennedy's new nutritional approach to the US chronic disease epidemics, because it's unfortunately all deuterium related, processed food, glyphosate, you name it. So practically what we share here is very valuable data, very valuable information. And I wanted to just make one correction. Yesterday, when I was making a note, I was saying deuterium is not, biology does not have deuterium. I was going to say biology does not have equilibrium in the proton tunneling system, simply because deuterons are actually discriminated from this entire process, and it's an electron-proton-based system, which, so thank you, Gábor. For 50 years, it actually carried us on, and it will go take us further. So thank you for your attention.